So in this recording, we're going to move away from molecules and start to think about how those molecules come together to form cells. So if we go back to our biological hierarchy, we've talked about atoms and how atoms come together to form molecules. We talked about our 4 biomacromolecules: carbohydrates, lipids, proteins, nucleic acids. And now what we want to look at is what is the unit that is our basic building block of life, that is the cell, and how do those molecules, those biomacromolecules, incorporate themselves, organize themselves to create this emergent property called life at a cellular level. Now, all cells are going to break down into 2 major groups called prokaryotic cells and eukaryotic cells, which we will delve into in great detail. But all cells are going to have a basic features that include a plasma membrane, which is going to be the membrane whose foundation is what's called a lipid bilayer, which you've incorporated now proteins and carbohydrates into. But this is going to be our defining barrier between the cell and the environment. The inside of the cell, a general term we can use is called the cytosol. That's all the living stuff, the living chemistry that's going on inside the cell. All cells have at least 1 chromosome, and that chromosome is going to be made of DNA. It's going to carry the information to make proteins that drive the life of that cell. And then ribosomes are found in all cells, and these are the site of protein synthesis. And we will go over these in greater detail later. This is our overview. So again, if we look at these 2 kinds of cells, these are not drawn to scale. You can see the scale here. Prokaryotic cells are relatively simple, and you're going to hear this again and again from me, while eukaryotic cells are very complex. They are compartmentalized. So let's take a look at some cells. These are beautiful pictures of fluorescent microscopy. This is, you use special labels to make special parts of the cell light up. This is the cytoskeleton of a eukaryotic cell. We can see a paramecium. This is under light microscopy. You can see this in the lab. Again, a eukaryotic cell, complex interior compartments. Plant cells. Here we are with the green chloroplasts where photosynthesis is occurring. You can see they're like bricks in a wall. This is a multicellular organism made up of many, many cells. Whereas then we have prokaryotic cells, bacteria. These, for example, have flagella, but they're much, much smaller. We want to keep that in mind. So as we talk about big and small, we need to review our metric system, and we want to keep it in mind what scale we're talking about when we talk about molecules and cells. And so what we can see here is this yellow area in the middle of this figure, which shows you where the cells are. And cells are going to range in size from 1 micron to 100 microns. So that's going all the way down from most bacteria being about 0.1 to 10 microns, and most eukaryotic cells being from 10 microns to 100 microns. Now, what is a micron? So we want to put that into context in the metric system, where most people are familiar with centimeters and millimeters. If you divide a millimeter 1,000 times, you get a micrometer, or a micron. And the symbol that we're going to use for this is the Greek letter mu. So where you have C for centi, M for milli, you're going to have the Greek letter mu for micro. We also call micrometers microns. It's 1/1,000,000th of a meter. Now, we'll also talk about nanometers, which is 1,000 nanometers per micron, and we'll use the letter N to represent that. Now, it's a fine time to go ahead and memorize then pico, femto, atto, and zepto at 10 to the negative 12th, 10 to the negative 15th, 18th, and 21st of a meter. Right? You'll also hear the term angstrom at times, and that is the diameter of 1 carbon atom is 1/10 of a nanometer. So these are the scales of micro and nano are the scales that we're going to be spending a lot of time with this quarter as we delve into the cellular world. Not something we can see with the naked eye, but something we have to be able to envision and imagine and remember what scale we're working with. Now, as I've mentioned before, there are 2 main types of cells, prokaryotic and eukaryotic. So bacteria and archaea are 2 domains, types of living things that are prokaryotic. Now, protists, fungi, animals, and plants, most of the things that you're familiar with and see in your everyday world, these are all made out of eukaryotic cells. So when we compare these 2 cells in a microscope slide, we can see these little purple dots all over this slide. Those, in fact, are bacteria. Those are prokaryotic cells. Whereas this big pink blob with a small, you can see the nucleus of that cell in the middle, that is actually one eukaryotic cell. So we can see immediately that the eukaryotic cell is much, much larger than the prokaryotic cell. When we look at these types of life, as I mentioned, bacteria and archaea, we can talk about the relatedness of all living things. All right? Eukarya is the domain in which all eukaryotic cells reside. So anything that's made of eukaryotic cells will be in this domain, eukarya. This is the highest level of taxonomy. This is the way we organize living things. Now, archaea and bacteria are both prokaryotic cells, and they have some differences that justify their being in two different domains, and we'll talk about that as the quarter progresses. But this idea of relatedness can be displayed in this phylogenetic tree. So this is a way to represent how things evolved through history. So at the top of this slide we have the present age, and we go back all the way down to the bottom of the slide at the origin of the Earth. That would be 4.5 billion years ago, or 4,500 million years ago. And so according to current understanding, life probably evolved somewhere between 3.8 and 4 billion years ago. And based on the evidence from current living things, we probably all evolved from one single event of life emerging. So as you look through history, then, you can see how prokaryotes, bacteria, and archaea diverged from our evolutionary line early on, but then all eukaryotes have a common ancestor. Right? And we'll talk about this more as we talk about evolution. But this does introduce the idea of taxonomy. And for example, this is our way to categorize a certain type of life, like the American black bear, Ursus americanus. So all the black bears out there are a type of life, right? But those black bears are more related evolutionarily to all the other bears than they are to anything else. And so we can group all the bears together into a genus, Ursus. Now, bears are all more related to each other than they are to other kinds of life, but you can walk your way up as you get more and more distant relatives, but still grouping them together. So we go from species to genus to family to order to class to file and to domain. And again, domain is going to be our most inclusive category, and in this case is eukarya. All things that are eukaryotic are in this domain. We're going to talk about this more as we talk about evolution, but I do want to introduce this now so people have some idea as we talk about different species. Now, again, prokaryotic cells tend to be simpler, single compartment, not a whole lot going on, compared to eukaryotic cells, which tend to be very complex. Again, these cartoons give you a good idea of how packed a cell is, but they are just cartoons. We'll talk about microscopy and how we derive these pretty pictures from some pretty interesting uses of microscopic technique. Now, again, before we leave this overview of cells, we want to remember that the major common barrier that's going to define a cell is the plasma membrane. This is going to be the barrier through which everything that comes into the cell must pass, and everything that exits the cell must go out of. In general, we see this as a lipid bilayer. So remember we talked about lipids, specifically phospholipids. Remember, phospholipids have a hydrophobic portion, these fatty acid tails, and a hydrophilic portion, that head group that is charged. It'll form this bilayer, and you want to think of it almost like a soap bubble. It's that thin. Now, physically, it's not necessarily very robust, but chemically, it's very robust. And we'll talk about that more as we get into our chapter on membranes. But we can see also that we've integrated proteins and carbohydrates into that membrane to make it much more functional. Now, that is going to be our delineating point. Inside of this is the living cytoplasm, and outside of this is the environment. And all cells are going to share that in common. All right, with that, then we'll move on now to looking at in greater detail each component of prokaryotic and eukaryotic cells.